Welcome back everyone to the Kingdom Zoology channel. So let's continue forward with our lectures of movement and locomotion. What we have done in movement and locomotion chapter till now, I'll just give you a quick recall. The muscles, which will be basically collections of bundles called fasciculi. Fasciculi will be bundles of individual cells called the muscle fibers. So here, this is a fiber, this is a fasciculus, and here is the entire muscle. So muscle is a collection of fasciculi. Fasciculi themselves are collections of cells called fibers. Now if you talk about the fibers, these are cells. What do the cells contain? Proteins inside them. What are the proteins of the cells called? Myofibrils. The protein rods in the cell are called myofibrils. And the next thing that I told you was that myofibrils themselves are made up of myofilaments. You can notice two types of structures here, one drawn in red color, other drawn in pink color. So the red color structures are the secondary or the thin myofibrils, and the pink ones are the primary or the thick myofibrils. So let me just label them again for you over here for our reference. These structures are the thin or secondary myofilaments, while the pink ones are the thick or the primary myofilaments. And as you might recall, what we had discussed in the previous video, that the thin or the secondary myofilaments are arranged like this, while thick filaments are over here in the center. So because of this characteristic arrangement, there are certain areas of the myofibril where thin and thick both filaments are present. So these areas will be dark. Then there are certain areas with only thin filaments, light. This also will be slightly lighter because there is only thick filament over here. Once again, thin and thick filaments overlapping each other, so dark, slightly lighter in between, and then once again what? Light over here. So the dark and light bands are basically these structures. These are our dark bands here, the areas with both thin and thick filaments overlapping each other. These are the dark bands. This is one dark band, dark band over here. The dark bands also have slight light area between them, and of course then there are complete light areas with only secondary filaments. So what we are going to do today is basically discuss the structure of subunits of the myofibrils. What are the subunits of the myofibrils called? They are called sarcomeres. So it's a very common misconception. Some people basically will tell you that sarcomeres are the structural units or the functional units of muscle fibers. That is technically wrong, because as you can see in this picture here, and as we discussed earlier, I'll just give you a quick recall in the written form also. Muscles are groups of fasciculi, which are themselves groups of cells or the fibers. The fibers contain myofibrils, elongated protein rods inside the fiber, and the myofibrils are made up of or consists of myofilaments of two types, the thin and thick. Thin secondary, thick primary. Okay, and it's basically the arrangement of these myofilaments. The arrangement of myofilaments in a characteristic pattern that we can see over here in the structure. The arrangement of myofilaments results in dark and light areas or dark and light bands on the myofibrils. So since each myofibril will be having the dark and light bands, myofibrils are the dark and light bands. The cells also seem to be having them. The fasciculi also will be seem to be having them. The muscles also will appear to be having the same dark and light bands. Okay. So here is our arrangement of various structures inside a muscle tissue. So what we'll be doing today is, as I told you, the myofibrils can be divided into structural as well as functional units, we can say, called sarcomeres. So that is what I was trying to tell you a while ago, that if there is any question or anybody who tells you that sarcomeres are the structural and the functional units of what? What is your answer? If the option says muscle fiber, is that correct? No. Because sarcomeres, the structures that we are discussing today, are the structural and functional units of myofibrils and not the muscle fibers. Because muscle fibers are cells. The cells are made up of collections of protein rods called myofibrils, and each myofibril has got myofilaments. The arrangement of those myofilaments results in the dark and light bands on the myofibril. So the repeating structural units of the myofibrils actually are what? Sarcomeres. Now what exactly is a sarcomere? Let's try to understand that in this structure here. In this structure we can notice that there are secondary myofilaments, the red color structures, and the primary myofilament, that is the pink color structure. Now in the structure of the sarcomere, in the structure of the myofibril, we'll notice that there are faint lines visible when you observe it under a microscope. There are very faint lines which are visible in the center approximately of what? The entire length of the secondary myofibril. This over here is an entire secondary myofibril. This is the entire secondary myofibril. So exactly in the middle of the secondary myofibril, there are faint lines that will be visible in the microscope. What are these lines? Over here also there will be one. On this side also, if we extend the structure further, there will be one similar line over here as well. Okay. So these are the faint lines which are again made up of what? Proteins. Okay. So what do we call these Z lines as? They are called as Z lines. There is another term for the Z line. It's also called the cross membrane. The Z lines are also called cross membrane, KRUSE. So in a myofibril, there are secondary myofilaments and then there are primary myofilaments. In the center of the secondary myofilaments, what do we have? The Z lines of the cross membranes. In a myofibril, faint lines called Z lines or cross membranes are visible in the middle or the center of the thin or secondary myofilaments. What are these lines called? The Z lines or the cross membranes in the exact middle of the secondary myofilaments. As we have seen in the structure here, once again, this is your entire secondary myofilament. In the middle, there is a Z line. Once again, the red line is the secondary myofilament, Z line. Secondary myofilament, Z line. Secondary myofilament, Z line. On this side also secondary myofilament, in the exact center there will be another Z line. The area between two successive Z lines, what is this called? This is a sarcomere. If there is another Z line somewhere over here, that's another sarcomere. This is one more sarcomere. This is one more sarcomere. So now tell me, can I say that sarcomere is a repeating structural unit of the myofibril? Yes, it is, because you take one sarcomere, second sarcomere, third sarcomere, connect them to each other. What do you get? The entire myofibril. Okay. So the area between two successive Z lines is called a sarcomere. Now if I basically, let's say, just choose one single sarcomere, I'm going to basically draw another structure now. This is one Z line, this is the other Z line, the area between them is the sarcomere. So let's just focus on this much structure now. Here is your one Z line, and here's your second Z line. I'll just try to keep it in sync with the upper picture, even though it's a highly zoomed out image, but still, this is one Z line. Somewhere over here will be the second Z line. I'm drawing the structure of an entire sarcomere in detail now. This is, let's say, your second Z line. Between two successive Z lines, what is there? A sarcomere. Now if you look at the upper picture, you will be able to understand between Z lines what do we need to draw. Here secondary myofilaments, here again secondary myofilaments, and in between them there is the primary myofilament. That is the structure of the sarcomere. So let me take you back here now. On this side there is a secondary or a thin myofilament. Here also one more secondary or the thin myofilament. Secondary myofilaments are called thin myofilaments, obviously because of their thin nature, right? And between them there is a thicker structure or a thicker rod, which is called the primary or the thick myofilament. Now look at this structure and compare it with what I've drawn over here. It's the same. Over there what we've done is two Z lines. What are these? Thin secondary myofilaments, in the center thick myofilament. So I've drawn the same structure over here. The Z lines, thin myofilaments and thick myofilament in the center. Thick myofilament in the center and secondary or thin myofilaments surrounding it. If this is one sarcomere, over here will be the next Z line. So as you know, this will be the entire complete structure of your secondary or the thin myofilament. On this side also one more secondary or the thin myofilament set. Over here one more primary or thick myofilament. So we can notice that this is the next sarcomere, and the structure will just continue. Somewhere over here will be another thick myofilament, then more thin myofilament, one more Z line. So that is how the sarcomeres will continue. On this side also, that's the entire complete structure of your thin myofilaments. Over here you'll have the thick myofilament, then another thin myofilament set over here, another Z line over here. So this is your sarcomere, 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 the repeating structural unit of the myofibril. Okay. In this structure, we can notice now in the sarcomere that there are thin myofilaments, there is a thick myofilament, and there are certain parts of the sarcomere where we have only thin myofilaments. There are certain parts of sarcomere where thick myofilament is overlapped on both the sides by thin myofilaments. Then in the center there is a slight part of the thick myofilament that is free. Then again thick myofilament overlapped on both sides by thin myofilament, and then once again only thin myofilaments. So then if we recall the same story that I told you the other day, if I put this sarcomere on a source of light and light is passing through the sarcomere and reaching your eyes, there will be different parts of the sarcomere giving different appearances because of the difference in the light absorbance that they will have. So if I divide the sarcomere now into different smaller portions, that is what I can actually do, the distinction between various portions of the sarcomere. So when we look at this structure now, if I talk about just this much portion of the sarcomere, this is the portion of the sarcomere which has got only thin myofilaments. Hence, light in appearance. This is the part of sarcomere with thin myofilaments overlapping the thick myofilament. So since the protein density is higher, thin and thick both proteins are there. So this has got what? Dark appearance. Because light absorbance in this area will be very high. So if light absorbance is high, this area would appear what? Darker. Look at this portion. Part of sarcomere with only thick myofilament. So once again slightly lighter. And then repetition here. What do you have? The overlapping of thick myofilaments by thin or the secondary myofilaments. And the last portion, that is once again this one over here. Only secondary myofilaments. So once again lighter in appearance. If we again go back to the top structure here, I told you that this will be light area, dark area, light area, dark area, light area. The entire light structure on the myofibril from here till here, this is called the light band. From here till here, what is this structure? The light band of the myofibril. This is one more light band of myofibril. This is one more light band of myofibril. So this opens up another information for us, that the Z lines are present in the exact center of what? Light bands. If this entire area is your light band, where's the Z line? In the exact center. If this entire area is the light band, where's the Z line? Exactly in the middle. That's another light band, where's the Z line? Exactly in the middle. Okay. So now if we see in the structure of the myofibril, this entire area is the light band. This entire area is the light band. So if I'm drawing one sarcomere from Z line to Z line, how much part of this light band is that? How much part of the light band is this? Half, because from here till here is the full I band. So how much portion of the light band belongs to one sarcomere? Half. Similarly, this entire structure is one light band, so how much portion of this light band belongs to this particular sarcomere? Only half. So if this is your entire light band, how much is this part? Only half of that light band. Here also, this portion is how much of the light band? Only half, because the full light band is this much. Okay, and this much portion of the sarcomere or the myofibril, we can say, is the dark band. तो ये वाला पोर्शन क्या है? डार्क बैंड. ये वाला पोर्शन क्या है? डार्क बैंड. ये क्या है? एक और dark band. So if you look at it carefully now, on the structure of myofibril, what do I see? Light band, dark band, light band, dark band, light band, dark band, alternating with each other. Let me basically just make it dark to help you understand the distinction. So in the structure of myofibril, I can see alternate. Here also is one more dark band. I can see alternate dark, light, dark, light, dark, light, dark, and then light again over here, bands. That's why they're called striated muscle fibers, because they've got striated myofibrils with alternating dark and light bands. So when I'm drawing the structure of one sarcomere, understand this here now. I'm drawing the structure of one sarcomere. How much portion of the I band is this? Half. How much portion of the I band is this? Half. And how much of the dark band is there in one sarcomere? One complete full. And you can notice that why is this portion of the sarcomere light? Because there are only one type of proteins here, secondary. If you look at this portion of the sarcomere, this is again light because there is only one type of proteins over here, secondary. But if you look at the central portion of the sarcomere, this appears dark because over here we have got both the types of proteins. In some areas on the periphery, and only one protein in the center, that is primary. So in the entire structure of the sarcomere now, there are half of the light bands on both sides, and in the center there is one full A band. Got it? So let me take you over here now. In the structure of the sarcomere, this portion is basically your half of the light band. Why is it light? Because there is only thin myofibrils. This again is light because there are only thin myofibrils, but I've told you that this is actually only half of the light band. This is also what? Half of the light band. Why are these halves of the light band? Because if there is one more Z line over here, that's your another arrangement of the same myofibrils. Okay, so this is the next half of the light band. So that will be now your entire light band. The Z line is bisecting it. So half of light band belongs to one sarcomere, the other half belongs to the next sarcomere. And this entire area is the dark band. Even though this is also not completely uniformly dark, there are some areas in it which will be more darker, these parts, because इधर क्या है, दोनों टाइप के प्रोटीन्स एक दूसरे को क्या कर रहे हैं, overlap. And there is a central area which is a bit lighter. Why? Because there is only one type of protein here, only secondary. So that is how a sarcomere would actually look like. Half of the light band, one full complete A band, and half of the next light band. So we can see in the structure that a sarcomere is made up of three parts. The three parts are half of one light band, complete dark band, half of the next light band. That is the composition of the sarcomere. Half of the light band, half of the next light band, and one full complete A band in the center. The light bands of the sarcomere are also called I bands or isotropic. Isotropic term means uniform. Because if you look at an entire light band, look at this structure here now. This is your entire light band. It's totally light, completely, uniformly light. Here also is one complete light band, completely uniformly light, except just a small faint Z line in the center. Here also completely uniformly light. But between them, what is there? Dark band. Between them, what is there? Dark band. But if you look at the dark band carefully, is it completely uniformly dark? No. If this is your dark band, these two lines are extra dark and the central area is a bit lighter. In the dark band, this area is extra dark, This area is also extra dark, but the central area is a bit lighter. Because of the arrangement of proteins, if you look here, what type of proteins are there? Both of them, secondary and primary myofilament. Over here only primary myofilament is there. So in the dark band, these two parts will be extra dark, while this area will be a bit lighter. That's why dark band is called anisotropic, that is non-uniform dark band. Light band is isotropic. Light band is isotropic, completely light. Dark band is anisotropic. Some areas are extra dark, some area is a bit lighter. The light bands are also called I bands or isotropic bands, uniformly light. While the dark bands are also called A bands or anisotropic. Non-uniformly dark bands, as an A band consists of two very dark zones called overlapping zones, in short, O zones. What are the overlapping zones of an A band? See, look at this structure now here. This is your dark band or the A band. It has got one portion here and one portion here, the extra dark areas. The extra dark areas are also called the O bands. Why O band? Because there is overlapping here. O stands for overlapping. The primary myofilament is overlapped by secondary myofilaments. The primary myofilament is overlapped by what? Secondary myofilament. So these are the two extra dark areas of the dark band, but in the center a bit lighter. Why? Because there is only one type of protein here. Which one? Thick or primary myofilament. The central area in the dark band, which is a bit lighter, is called the H zone. The word H here stands for Henson, the scientist who explained this structure. So if you look at the I band, uniformly light, but if you look at the dark band, it is not uniform. There is a dark area, ozone, slightly lighter area in the center, H zone. That's why dark band is anisotropic, not completely or uniformly dark. It's dark, lighter, dark. While I bands are completely uniformly light only. So the dark band is also called A band or an isotropic band because it consists of two very dark zones called overlapping zones, where the primary myofilaments are overlapped by the secondary myofilaments, and the central area of the A band is a bit lighter in appearance, as it has only what type of protein? प्राइमरी मायोफिलामेंट्स इन इट, सेकेंडरी तो नहीं है सेंट्रल एरिया में. So this slightly lighter area is called the H zone or the Henson zone. So this is the composition of the dark band, and here is the structure of your one sarcomere. Why does it have this appearance? Light, dark, light, dark, light. Because of characteristic arrangement of myofilaments, secondary and primary. So that is what the structure of sarcomere is, guys. Sarcomere is basically what? The repeating unit of the entire myofibril. And then the many myofibrils will be present in a single muscle fiber. So I think you understood this. I'll basically give you one more view over here to actually give a better understanding finally as a revision, as well as basically to give you a macro level view of the entire muscle fiber or the cell. Before that, I'll mention one more thing. We can say that a sarcomere is made up of, what is the sarcomere made up of? Half of one light band, complete dark band, half of the next light band. So I can say that a sarcomere is made up of, light band is also called I band, so I by 2. Dark band is called A band, light band is called I band. So a sarcomere is made up of half I band, one full A band, and half I band. This is Z line, Z line. The area between successive Z lines is called sarcomere. What are these proteins that I'm drawing now? Secondary or the thin myofibrils. And what is this that I've drawn in the center? Primary or the thick myofibril. So that's another view of the sarcomere. And let me basically highlight the areas also for you. What is this portion? Light. What is this portion? Light. So how much of the I band is this? Half. And how much of the I band is this? Half. With this being your Z line, this also being the other Z line. And if you talk about the dark band, what is this portion? Extra dark. Why? Because how many types of proteins are there? Both. Primary as well as secondary myofibrils. But what about the central area? That's a bit lighter, because there is only one type of protein. What is that? Primary. So ozone, ozone, H zone, and the entire structure is basically what? The A band. So that is another view of a sarcomere. Why did I draw it again? Because I wanted to show you how the sarcomeres can be called as repeating structural units of the myofibril. So what am I doing over here is arranging sarcomeres one after another. So this is basically sarcomeres repeated continuously. Choose any sarcomere. What is this in the center? One full A band. What is this? Half of the I band, because full I band is this much. What is this again? Half of the I band, because full I band we can notice is this much. So each sarcomere has got halves of the I bands and one full A band. So the structure that you're looking at in front of yourself right now is a structure that is made by joining a lot of sarcomeres together. When I join a lot of sarcomeres together, this is a sarcomere and this is a myofibril. Here's a sarcomere, and this entire structure is a myofibril. And you know that myofibrils are the building blocks of muscle fiber. So these are, let's say, various myofibrils. How many of them? 4. So in this picture, what is this that you're looking at? A myofibril. This is a myofibril. Second myofibril, third myofibril, fourth myofibril. Now do one thing. Add some cytoplasm around it. Around them. Add a few nuclei. And make a cell membrane. Sarcolemma. So what is it that I've drawn here? A muscle fiber. So now if we recall right from the beginning, a muscle fiber contains units called myofibrils. The myofibrils themselves are made up of what? Myofilaments. Thin myofilaments, thick myofilaments, which are responsible for the alternate dark and light arrangement. And if this is one muscle fiber, that's another muscle fiber. That's another muscle fiber. What is the connective tissue between these muscle fibers? Endomysium. So this here is your muscle fiber. And if this is one muscle fiber, this entire thing is a fasciculus. Okay. And if this is one fasciculus, let me add one more fasciculus here, with the binding material between the fasciculi being perimysium. So now this entire structure is a muscle. आ गया समझ? ये पूरा स्ट्रक्चर मसल है, जो किसका बंडल है? फेसिकुलाई का. हर एक फेसिकुलस किसका बंडल है? मसल फाइबर्स का. एंड लेट्स जूम इन. हर एक मसल फाइबर किसका कलेक्शन है? मायोफाइब्रिल्स का. और हर एक मायोफाइब्रिल किसका बना हुआ है? अलग अलग टाइप के मायोफिलामेंट्स का, जो रिस्पॉन्सिबल है किसके लिए? फॉर द डार्क एंड लाइट बैंड्स. दैट इज योर एंटायर स्ट्रक्चर ऑफ द मसल, which is made up of fasciculi. Each fasciculus is made up of fibers. हर एक फाइबर के अंदर ये जो लाइन्स हैं ये कौन है? मायोफाइब्रिल्स. और हर मायोफाइब्रिल किसका बना हुआ है? मायोफिलामेंट्स का. So look at this now. Is your muscle looking striated? Stripes दिख रहे हैं? Yes. तो बस हो गया, यही striated muscle है. And that's it for today guys. अगली क्लास में हम लोग क्या करेंगे, विल बी बेसिकली टेकिंग अप डिटेल्ड स्ट्रक्चर ऑफ द इंडिविजुअल मायोफिलामेंट्स ऑल्सो. प्राइमरी मायोफिलामेंट किस चीज़ का बना हुआ है, सेकेंडरी मायोफिलामेंट्स किस चीज़ का बना हुआ है. सो विल कंटिन्यू विद दैट इन द नेक्स्ट क्लास. Thank you for joining, and of course keep studying, keep sharing the links about the channel with your friends, and I'll see you in the next session.